invite. I'm really excited to be here and to share with you the work that I did throughout my PhD at Emory University in Hayden Kissig's lab. So this is all published work. It was published around December of this past year. And yeah, I'm excited to show you guys and walk you guys through the data. I think it's a great opportunity as well to showcase maybe some things that got hidden in the supplementaries, and they're just as important, but journals, they have some limited space and we can't talk about everything. So hopefully I can show a little bit more of what we thought was important from our work. So our work entitles Differentiation Fate of Stem-like CD4 T cell that controls immunity to cancer. And so how this first started as a project, and this is something that we can't always showcase in a paper, is that it started from previous work of a PhD student in Hayden's lab. Her paper was really about where CD8 T cells and dendritic cells co-localize within a tumor tissue. And so what you're looking at here is a tumor of a kidney cancer patient. And this is an IF image of antigen-presenting cells, right, in green with MHC class 2. We think these are dendritic cells. And they really cluster together in these so-called tumor niches. And this is extremely important for the survival, right, of patients. Patients that are well infiltrated with CD8s and co-localized with DCs within their tumor, they respond really well to therapies, they survive really, really well. And when I came into the lab, we noticed that within these niches, there were also a lot of CD4 T cells that co-localized also in that same environment. And so we wondered what those CD4 T cells were, and if they in fact played a positive or negative role within the niche itself. And so, just to give a little bit of an intro to CD4s, I think we all know that CD4s are not as simple as CD8s. They come in many different flavors, right? They're very heterogeneous. And so when they undergo their priming and activation, depending on the cytokines that are secreted, usually by the innate immune system, they choose a certain transcription factor and program to undertake. And all of these CD4s really have a different function within the immune system, right? If we think about type 1 immunity, we generally associate Th1 and TFH CD4s. If we think of type 2 or type 3 immunity, Th17s or Th2s are usually our effectors. And so this gives the immune system really the flexibility of targeting different types of pathogens and different immunity based on the effector functions of each of these CD4s. And so what we wondered was, well, what is the differentiation, right, that CD4s are actually taking within the cancer response? And so, from the literature, if we look into it, there's a lot of different messages from CD4 T cells from patient data. There's a lot of support that having helper T cells within their tumors is beneficial for anti-tumor responses. We know that CD4 T cells can help the innate immune cells to have better functions. We know they can actually help CD8 T cells through cytokines and other things, maybe CD40 ligand. They certainly can help B cells if they produce better antibodies. And there's some talk that there might be cytotoxic T cells, CD4 T cells that can actually direct lyse cancer cells. But there's another side of the literature that suggests that all of the CD4s that go into the tumor response are potentially suppressive and or dysfunctional. And so the correlative data from the patient survival analysis doesn't really answer how we have dysfunctional T cells, but we have positive responses. And so we really wanted to take all of this into our question and determine what says that a naive CD4 will undertake a program that will contribute or suppress the tumor response. And so we took a little bit of a broad approach at first using our human samples. At Emory, we're very lucky to collaborate with a lot of great surgeons. This is mostly what I'm going to talk about. It's going to be kidney cancer, but we also saw the same things with prostate, bladder, and even brain metastases. And so the first thing we did is we looked at within the CD4 T cell compartment in a kidney tumor, there's a very large population of PD-1 positive CD4s. And PD-1 in this case suggests to us, given that we don't have antigen specificity, that these cells are antigen reactive, right? They're currently responding to something as PD-1 dictates. It's dependent on TCR signaling. And so we underwent single cell RNA sequencing within these cells to see the phenotype of the CD4s within these kidney cancer tumors. So these represent multiple patients, and they're all within the single cell UMAP. And we can tell that there's three main populations that really come across within this kidney tumor. Again, these are all antigen reactive, so they're all PD-1 positive. What we can tell is there's this cluster that is your classical Treg cluster with the FOXP3 transcription factor. There's a cluster that is characterized by the transcription factor EOMs, and these have been kind of noted as non-canonical Th1s from other research. And then there was a very large cluster where the majority of the cells really landed, and these were characterized by the TCF1 transcription factor. And we know from the CD8 literature, both in exhaustion and even in cancer, that this transcription factor is very, very important for the maintenance of this so-called, precursor or stem-like CD8 T cell population. And in fact, if we place the signature of a human stem-like CD8 from tumors into this UMAP, they enrich, the same program enriches within this cluster 3 of the cells that do express that same transcription factor, suggesting the program applies to maybe these CD4 T cells that are in the tumor. And so a little bit more on these cells, right? They're all antigen reactive. They have markers of antigen experience. They have PD-1, CD69, chemokine receptors that suggest they've migrated to the tissue. But they fail to express really a lot of effector molecules and or transcription factors that say that this population is a canonical CD4 that we know of, right? They don't express any effector molecules here, but they do express a lot of co-stimulatory signals and pro-survival cytokine receptors, including the IL-7 receptor. And so we were curious to see this in a lot more patients and do it by protein by flow cytometry. So what we did is we took kidney cancer patients. You'll see the tumor at the top. And we actually were lucky to find many patients that we got non-metastatic tumor draining lymph nodes to actually look to see if there was a difference in the populations within these tissues. And so what you can see here at the top within the tumor, the phenotype of the CD4s that are PD-1 positive and are not naive, we have that same Treg population that we saw within the single cell RNA-seq. We have that EOMs population, that's about 20% of the CD4s. We get practically no Th1 response across these patients, and then no TFHs within the tumor itself. But what you can tell is about 50% of the cells, right, express this TCF1 transcription factor, but they fail to express a canonical transcription factor that has defined lineages of CD4s for years. And it's a very, very similar story within the tumor draining lymph node. And this is about 150 kidney cancer patients. And what you can tell is this population really dominates both the tumor and the tumor draining lymph node response. And what was really interesting is this population is activated, but fails to acquire or go through a canonical lineage. And so we wanted to look a little bit further into the functional aspect of this population. And so we characterized a lot of different functional or surface markers that we could use to sort these cells out and actually address their functional capacity. And so we came up with 3 different markers that really define these cells. CD26, also known as DPP4, the IL-7 receptor are very highly upregulated within this population, and then they're negative usually for CD39 or CD38. And so we can sort these populations and place them in culture under different conditions. And so we can put them under unstimulated or bead stimulation. This is just your normal CD3, CD2, CD28 stimulation with the additional cytokines that usually polarize cells towards different lineages of CD4s. And so what we find if we place these TCF1 positive cells under unstimulated conditions, they survive really well and they maintain their undifferentiated state. They're still PD-1 positive, they retain CD26, the IL-7 receptor, but they do not upregulate any transcription factor that's associated with a lineage. If we add TGF-beta and bead stimulation, these cells are very capable of undergoing many rounds of division and also upregulating the FOXP3 transcription factor, which is associated with that TGF-beta polarization. In strike contrast, though, if we place the effectors, also all the CD39 positives, if we do individually Tregs and or EOMs in each stimulation condition, these effectors have lost the capacity to proliferate, right? They maintain their phenotype. So if we originally had 20% Tregs in this population, they maintain that 20%, the rest are EOMs, but they failed to, one, change their differentiation state, and two, they gave up their proliferative capacity. And we can do this with many conditions, right? If we add IL-12 to this bead stimulation, we call this a Th1 stimulation, these cells now instead upregulate the TBIT transcription factor and fail to upregulate FOXP3. The effectors, same thing, have lost the proliferative capacity and they do not become Th1s just randomly in the dish. And we can do this with TFHs, EOMs, and many other conditions. And so it seems like this TCF1 population, regardless of the stimulatory condition that we place it in, not only is it capable of undergoing many rounds of division, but it's also capable of undertaking canonical programs based on whatever cytokines we place them in. And so we said, well, maybe we're giving too much stimulation with CD3, CD2, CD28, and maybe this is not what's happening within the tumor itself. So we did a more physiological stimulus to see if these cells were in fact capable of proliferating with antigen-presenting cell-derived, both TCR and co-stimulatory signal. So how we did this was we sorted each one of these populations from primary kidney tumors, and we also sorted patient-matched tumor dendritic cells. So these dendritic cells, both dendritic cells, CD11C positive, DR positive, came with the peptides that they were presenting from that patient. We didn't add any peptide. We just placed a co-culture of either the TCF1 positive cells with these APCs or the effectors with these APCs. And we culture them for 5 days with a little bit of IL-2 that is not sufficient by itself to induce proliferation. And what we find is that these TCF1 or CD26 positive CD4s are capable of undergoing many rounds of division when placed with these matched dendritic cells, whereas similarly to the beads, these CD39 positive effectors are not capable anymore of undergoing rounds of division. And so we in fact have this really functional but undifferentiated CD4 T cell within these patient tumors. And so if we come back to our model from the beginning, right, where I said CD4s are really heterogeneous and choose to normally take, in response to a pathogen, a certain effector program, this is in fact not really the case what's happening in cancer. You know, it looks a little bit more like this. It looks like we have a big population of maybe what we're calling stem-like, and I'll confirm that with some of the mouse data in the future, of these TCF1 positive CD4s that are really functional. They can both proliferate and they seem to be able to undertake pretty much any canonical lineage, but in the cancer response, based on what we find in the humans, it's really undertaking a Treg or this EOMs phenotype. And so this is what's happening in the patients. We're taking it out in vitro, right? And so it could happen, but is it actually happening, right? Are these cells antigen specific? Is this, TCR dependent? How does this cell decide whether to acquire a Treg phenotype or a functional differentiation phenotype? And how does this impact the anti-tumor response? Why would we care? All right, and so all of these questions, really, we went into mouse models to try and address how this population really impacts the anti-tumor response. And so what we did here is we acquired a prostate cancer cell line, and we put in the LCMV glycoprotein. This is full-length GP. into this prostate cancer cell line. This is a very slow growing cell line, which was really important for us to determine the kinetics of differentiation from naive CD4s all the way to 5 weeks later, what did they become? And so the beauty of this model is that we can also address antigen specificity based on CD4 tetramers that we have for LCMV. And so these GP66 are antigen-specific CD4s that we can find in specific to the tumor, that we can find both within the draining lymph node and the tumor at different frequencies. What we first found is that there's an incredible accumulation of these antigen-specific CD4s within the draining lymph node, and in fact, they have a hard time really migrating to the tumor. If we look at the phenotype, it really recapitulates what we found within these kidney cancer patients, right? We have a small proportion of cells that are differentiating into Tregs within these draining lymph nodes. We have very few Th1s that ever come up. This is 5 weeks after inoculation. And we do have a population of TFH cells that does develop within the draining lymph node. Within the tumor itself, at least in the mouse, it's a lot more pronounced that it goes towards a Treg differentiation trajectory. So over 70% of the cells within the tumor itself really go into a FOXP3 positive state, but we still maintain this TCF1 population that is undifferentiated within the tumor itself. And so what's really apparent is that even within 1 week after tumor inoculation, this TCF1 positive population is really the first one to come up. Again, they're antigen specific. Within 7 days, they're there within the draining lymph nodes, and they're maintained. Their numbers-wise, they stay really, really consistent throughout the 5-week response. Within the tumor, it's a little slower. We start seeing cells around 2.5 weeks in, and again, over time, we get an accumulation of differentiation towards that Treg trajectory. And so a little bit more on these cells, we can stain for markers of both activation and proliferation. Again, these cells are all PD-1 positive. They are consistently proliferating. This is 5 weeks after inoculation. About 20% of them are continuously proliferating within this draining lymph node. They don't express marks of other transcription factors that suggest they would be a different lineage. So they're not Th17, they're not EOMs. They do express very, very high levels of TOX, which we know is essential to maintain that stem-like population in CD8s. They express a lot of co-stimulatory markers, CD226 and SLAMF6, and a population of them do have marks of migration and probably something that is required to migrate to the tumor. And within the tumor, this phenotype doesn't change very much. They still maintain their PD-1 positive activity. They're still somewhat proliferating. They're TOX positive, and they maintain this SLAMF6 TCF1 positive state without the need of upregulating effector molecules. They maintain that undifferentiated state even within the tumor. And so they activate fine, they're proliferating, they accumulate within these tissues. They are antigen specific, but they fail to acquire these canonical helper programs. Instead, within the tumor, and we saw this, there's an accumulation of Treg differentiation that happens within these tumors. And so our next question was, well, are these cells just completely dysfunctional? Some people have called them anergic. So we were wondering, are these cells in fact dysfunctional because they got primed in an anti-tumor response, or are they just locked into this Treg differentiation trajectory because they got primed in a cancer response? And so to address these questions, we put in tumor-specific CD4s, and so we put in SMARTas. So these are TCR transgenic CD4 T cells that are specific for the LCMV glycoprotein. So we put them in day -1, we inoculate with tumors, and we wait for 5 weeks for these CD4s to undergo their normal differentiation trajectory, right? They will acquire that TCF program. Some of them will go to the tumor and acquire that Treg program. And so what we did then is sort the TCF1 positive lineage negative SMARTas from the tumor draining lymph node 5 weeks after inoculation. And we put them into a mouse, a naive mouse, that then received LCMV Armstrong. And so this LCMV Armstrong has the shared glycoprotein that these TCF1 positive cells have been seeing for 5 weeks. The question being, can these cells from a tumor draining lymph node that have been seeing the same antigen for 5 weeks in a mouse that now is receiving an infection, can they change their differentiation trajectory and undergo normal Th1 or TFH differentiation as you would expect in a viral infection? And so we looked 8 days later, and these cells, I think we put in as little as 300 cells, and we can find them in this naive mouse, indicating that one, they're very functional and capable of proliferating, expanding, and repopulating the population. And we can compare their differentiation state to the endogenous GB66 response from this naive mouse. And when you look at the differentiation trajectory of these smartest, these cells were capable of upregulating TBET, right, the Th1 transcription factor. They certainly did not become Tregs in this naive mouse. And I'll show you in a second, these TCF1 positive cells are actually TFHs. They fully upregulated BCL6. And this was true in every single tissue that we looked at, in the spleen, non-secondary lymphoid tissues or peripheral organs like the liver, and even we found them within the blood. And again, these TCF1 positive cells that were within this infected mouse fully upregulated BCL6 and really became TFHs. And so these cells are anything but dysfunctional, right? They are capable of undergoing massive proliferation. They can change their differentiation trajectory and really acquire canonical programs when the right signals are present. And so our question then was, well, if they are capable, then what is regulating their differentiation in cancer, right? Because they're not doing it in the anti-tumor response. And so we first thought, okay, maybe it's checkpoints, right? They have a very, very high level of PD-1 from the very beginning, and PD-1 regulates the effector differentiation of CD8 T cells. And so we thought perhaps, it's very similar, and perhaps if we block PD-L1, we can override this program and push them towards a more beneficial state. We still use this TRAMP-C1 model, and as note, this model is refractory to PD-1 therapy, but we also did it in 2 models that are responsive, and the results were very, very similar. And so the setup here is we inoculate with tumor, we wait for about 2 to 3 weeks for this tumor to be to establish, and then we treat with anti-PD-1 therapy for about 2 weeks. And what we found is when we look at the phenotype of antigen-specific CD4, so these are all gated on GP66, in untreated, it's just what I've told you, very little Th1s, a population of Tregs within the draining lymph node that is massive within the tumor. And then after PD-1 therapy, although we got enhanced proliferation of these cells maintain the same phenotype that they had in an untreated mouse. And so it seems like PD-1 is capable of restoring somewhat the proliferation of these antigen-specific cells, but it's just restoring the proliferation of what's already there. It's not changing the differentiation state of these antigen-specific CD4s. And so we thought, okay, well, what is the biggest population of PD-1 positive CD4 T cells within this cancer response? And in fact, if you look at bulk CD4s, not just antigen specificity, Tregs are a massive population of CD4 T cells that is both within the drain lymph node and the tumor. That is just not the case within any infection that you look at. And so perhaps having that large population of Tregs from the very beginning is really limiting the stem-like CD4 undergoing proper differentiation trajectory. And so how we did to address this, we acquired FOXP3 DTR mice. So with these mice, you can selectively deplete Tregs using diphtheria toxin. So how we did this is we inoculated either wild type or DTR mice with our prostate cancer cell line. We waited for 3 weeks for these tumors to be established, and then we treated with DT to selectively deplete Tregs. And what we find is that in untreated, right, just as I've shown you, GP66 CD4s, they're either Tregs or TCF within the draining lymph node, no Th1s. That changes dramatically 5 days after depletion of Tregs. The stem-like CD4, and I'll confirm this for you in many ways, gives rise to these Th1 populations that are within this draining lymph node, and that's also true for the tumor. And so we get a huge change in the differentiation trajectory of these cells right after Treg depletion. And this is really consistent even 25 days after the initial depletion and when the Tregs do come back. So this is the DREG model of FOXP3 depletion, and Tregs do come back, But now, once the differentiation trajectory has gone to Th1s, it seems that it's able to self-sustain itself, both within the draining lymph node and in the tumor, which was really interesting to us as well. And these cells are fully becoming Th1s, right? It's not just the upregulation of T-bed. They're not retaining TCF1. Now they're fully downregulating this stem-like program, and I'll show you a little bit more in a second. And very importantly, they're maintaining their numbers. So although they are differentiating and we're getting a huge spike of this Th1 population, we're also maintaining our numbers of stem-like cells, really suggesting that they're both undergoing proliferation and differentiation simultaneously. To get a little bit more about the changes that is undergoing right after Treg depletion, we did single-cell RNA-seq within both untreated and Treg depleted mice. So you can tell there is the stem-like population here in green, some TFHs similar to what we saw by flow, Tregs, and then this orange cluster, which is what I really want you to focus on, is that Th1 population that is arising pretty much exclusively in that Treg depleting condition. And what's important is that the stem population itself is really undergoing changes after Treg depletion, right? It's maintaining its score of stem-like, right? They're still tox positive, they're still TCF1 positive, and all the things associated with that program. But they're actually undergoing certain changes that suggest transcriptionally they're ready to undergo Th1 differentiation, right? All of these genes here that are upregulated in Treg depletion versus untreated are genes that really transcriptionally are needed to undergo Th1 differentiation. KLF2, all the LGALs and things associated with Th1s. And in contrast, those in the untreated draining lymph node are more Treg prone for differentiation, which was really, really interesting. And if we look a little bit more at the relationship between the stem-like population and the Th1 cluster, within that Treg depleted condition, we can tell that pretty much every clone that was in the top 10 expanded TCRs within the Treg depleting condition has both a stem-like population and a Th1 population. And if you look at all of the TCRs within that Th1 population, there was over 90% overlap with the stem-like CD4 T cell, really suggesting that they are in fact the precursors of Th1s that are arising after Treg depletion, which has been a subject of debate for a long time. And so we thought TCRs weren't fully enough to convince us that this was in fact real. We wanted to do a functional retransfer experiment to really, really show the relationship between these cells. And so how we did this was we acquired 45.1 SMARTas. Again, these are TCR transgenic CD4s, specific for the glycoprotein that is in this TRAM tumor. We waited for 3 weeks for these tumors to be established, and we sorted either the stem population from the draining lymph node or the Treg SMARTA population that develops within this draining lymph node. And we asked if we put them into a tumor-matched mouse, and we selectively deplete Tregs from this mouse, then can they give rise to that Th1 population? And what we find is that in untreated mice that do not get Treg depleted, these stem-like CD4s are capable of restoring that stem-like population within the draining lymph node and also giving rise to the iTregs that get there within the draining lymph node. So we're also showing that they are the precursors to the effectors, Tregs, that are within the draining lymph node in wild-type mice. When we look at a mouse that got Treg depleted from this endogenous population, we can tell that these SMARTas underwent Th1 differentiation, whereas the Tregs that got transferred maintained their phenotype. Really, really saying that this TCF1 stem-like CD4 is in fact the precursor to the Th1 population that's coming along. And this was true also within the tumor. It's very early of a time point, so in wild-type mice, they haven't migrated. This is certainly enhanced when you deplete Tregs, and the ones that made it to the tumor retain that Th1 phenotype, whereas the Tregs only retain their Treg phenotype, really saying that the stem-like population is in fact the precursor to the emergent Th1 cells. And so that's all great, but why do we care, right? How does this actually impact the anti-tumor response? Does it matter that a stem-like population chooses to become a Treg or chooses to become a Th1 cell? How does this functionally impact the anti-tumor response? And so first, I'm going to give an example of a treatment that we usually give to patients, and it's usually to target Tregs. So very similar to what we did in our mouse data, patients get CTLA-4 all the time. And what we did here is we got two clones of CTLA-4, one clone that is not Treg depleting, so it's simply blocking CTLA-4, and one that is Treg depleting. And very similar experiment, we wait 3 weeks and we treat with either one of these clones and we assess the phenotype of the CD4s. And what we find is that it's only within the Treg depleting clone that we get a change in the stem-like differentiation population towards a Th1 state. The blocking clone itself is not sufficient to change the differentiation state of these cells. They stay locked in and they continue Treg differentiation. And so CTLA-4 alone is not the only thing that is suppressing the stem-like population from undergoing this Th1 differentiation. And what's important from this is that it's only when the stem-like population changes to a Th1 response that we get anti-tumor immunity. This is also true within Treg depletion, as it would be no surprise to everyone. So we can argue that there's two states really within this draining lymph node response that we have described. We've described this kind of restricted state that is really a poor anti-tumor response, right? At the end of the day, the cancer wins, where we have a large population of stem-like CD4s that are really getting polarized towards this Treg state by a big natural Treg population. And then we have this, what we're calling an active response, where the stem-like population is actively giving rise to a Th1 response. And it seems like in this case, we get effective anti-tumor immunity. We could argue that effective anti-tumor immunity here could be because of three different things. One, the removal of Tregs itself is causing systemic inflammation, right? And it's independent of the conventional CD4s that are present. Tregs have many, many different functions, and so we could argue this is completely independent of the stem-like CD4. Number two is that the Th1s that are being developed have direct cytotoxic capacity, right? And so now we're getting a huge population of cytotoxic CD4s. Maybe they directly are going to kill the cancer. Or number three is that the differentiation of stem to Th1 is required to induce a better response towards through a tertiary mechanism. And so we can test these hypotheses pretty simply. The first hypothesis was conventional CD4 help would not be required, is all Treg dependent. And so how we did this was a simple full CD4 depletion using the GK1.5 antibody. And so we inoculate with tumor, we wait for 3 weeks, and we deplete all CD4s, including Tregs, prior to administering diphtheria toxin to a FOXP3 DTR mouse, and we observe the anti-tumor response. And what you can tell here is that when we deplete Tregs, but we also deplete the conventional CD4s, the anti-tumor immunity goes away. And so conventional CD4s are required for this anti-tumor immunity. Number two was that Th1s have direct cytotoxic capacity, right? So in order to test this, we depleted the other major T cell that is required for usually direct killing of cancer cells. So we depleted CD8 T cells using an anti-CD8 beta antibody 3 weeks after inoculation. And what we find is that if we Treg deplete and also deplete CD8 T cells, although conventional CD4s are undergoing Th1 differentiation, this is not sufficient to have a good anti-tumor response. And so you need both, right? You need the conventional CD4 T cells, and you also need the CD8 T cells in order to have a good anti-tumor immunity. So that leads us to our third hypothesis, which was either the stem-like population itself or their differentiation to Th1s is required to induce a better response. That clearly by this is CD8 T cell dependent. And so our next question was, well, how is this happening? Right, and so we did this experiment to try and address how conventional CD4s are helping the CD8 T cells. So we inoculate FOXP3 DTR mice, we wait for 3 weeks for the tumor to be established, and we either deplete Tregs using diphtheria toxin, or we fully deplete all CD4s using our GK1.5 antibody. And what we can tell is that we can look at the antigen-specific CD8 T cells, particularly within the draining lymph node. And this is work from our previous lab, Natalia, who I actually think is in the audience, and many others that have now shown that antigen-specific CD8 T cells within the draining lymph node in cancer only undergo stem-like differentiation. They fail to acquire an effector program, and it's only after they've migrated to the tumor that they actually acquire their effector capacity, right? It's kind of a two-step process. In the draining lymph node, they prime, they activate, but only acquire this TCF1 positive state that fails to upregulate a lot of effector molecules. What happens after we simply Treg deplete is we're changing this process completely. Now we're getting stem-like differentiation of CD8 T cells that are full-on effectors. They're really, really proliferating, and they're acquiring granzyme B and all the things associated with a very, very good effector program. This is only 5 days after initial Treg depletion. If we fully deplete all CD4 T cells, so Tregs and the conventional CD4s, this differentiation trajectory goes away within the draining lymph node. We still get a higher proportion of cells that are proliferating, so we get more numbers, but we failed to induce that effector differentiation within the draining lymph node itself. And that translates to a lot less effectors migrating to the tumor, and it translates to not having that good anti-tumor response that we saw before. So really having, and this is very, very similar, we did this in 3 or 4 different models, and in all the models, conventional CD4 help was required to induce effector differentiation within the draining lymph node of CD8 T cells. And it's not just a change of acquiring granzyme B. There's a lot of changes that goes on in these antigen-specific CD8s within the draining lymph node. They put up a lot of cytokine receptors associated with the Th1 response, so interferon gamma. They upregulate transcription factors also associated with better effector programs like TBET. They downregulate EOMs. And even the stem-like itself, like the stem-like CD8 T cell itself, upregulates things already that are associated with effector differentiation in comparison to those cells without CD4 T cell help. They just have very upregulated stem-like programs. So all the TOX, ID3, WNT, these are all things that really, really sustain that TCF1 program that stem-like CD8 T cells have. And so when we Treg deplete and we have CD4 T cells, these cells are already prone to undergoing effector differentiation, both transcriptionally and by protein. And if we compare that stem-like population between completely CD4 T cell deplete and untreated, although we have a lot of differences in proliferation, that core stem-like program is maintained, and we have no difference transcriptionally related to effector differentiation. And so we can update our model a little bit more in this restricted program, where now we have a huge stem-like CD4 population. It's undergoing ITRA differentiation. And additionally, as Natalia from our group and very many others have shown, is that we have a big population of stem-like CD8 T cells within the draining lymph node. But with limited CD4 help, they're both pretty restricted. And it's not until these cells get to the tumor that they undergo effector differentiation. In strike contrast, though, when we have an active response and we have a Th1 population that arises, some help happens here that now we have stem-like CD8 T cell differentiation towards effectors, and we get an active anti-tumor response. And so we wanted to address what this was and what help was actually required or sufficient to really overtake that restricted program. And so we first said, if we put Tregs back in the system, because all of this that I've just shown you is without Tregs in the system, right? It's Treg depleted. So we said, if we put Tregs back in the system and we force stem-like CD4 T cells to undergo Th1 differentiation, would this be sufficient to override the restricted Treg program and promote stem-like CD8s to undergo effector differentiation? How we did this, we tried to address it many different ways, but how we came up to it in the paper was we made an enforced Tibet stem-like CD4. So we can essentially enforce the Th1 program in stem-like CD4 T cells that are antigen specific. We did this through a bone marrow chimera system that with time, I'm not going to go through all of it, but essentially we can make bone marrow chimeras with this construct. So this is a tetracycline dependent construct. And so in the presence of doxycycline, mice that have the tetracycline promoter can go and bind and basically overexpress TBET in the cells that we inoculated. And so these mice are smartest, so they're TCR transgenic for the glycoprotein, and they're also TET-dependent enforced TBET. And this is just to show you that the model works, right? And so wild-type smartest, usually they only become either stems or Tregs. Our TET-on TBET smartest in the presence of doxycycline, we can enforce them to be Th1s, and I'm not showing it here, but this is full-on Th1s. They can produce interferon gamma, they fully downregulate TCF1, etc. But these cells are not broken, right? Without the presence of doxycycline, they become stem, they differentiate into Tregs, and all the things normal wild-type smartest would do. Right, and so this is the experiment. We transferred in a very small amount of smartest in these mice that have the tetracycline activator. And so wild-type smartest or TET-on TBET smartest were transferred into a mouse that received a tumor. We waited for 3 weeks for these cells to expand and undertake their normal program within the draining lymph node and tumor. And then we gave doxycycline in the presence of PD-1. And now we're looking at the endogenous GP66, so antigen-specific CD4s from this mouse, right? Did the presence of TET-on or wild-type smartest influence the differentiation state of antigen-specific CD4 T cells in this mouse? And so in wild-type smartest, or after transfer of wild-type smartest, our endogenous CD4 T cells did not change, right? We have a big presence of TCF1, we have some differentiation of Tregs within the draining lymph node. Very few cells are producing interferon gamma, as you would expect in a non-Th1 differentiation response. But in the presence of TET-on TBET smartest, now the endogenous population is also coming along, and it's also undergoing Th1 differentiation within the draining lymph node and starting to produce a lot more interferon gamma and IL-2 within this tissue. This was also true within the CD8 T cell response, right? We're changing the CD4s. Now, if we look at the antigen-specific CD8s, we're also getting effector differentiation of these CD8 T cells within the draining lymph node, which translates to a more effector cells within the tumor itself. And as I mentioned in one of the first slides, is that this tumor itself, the TRAMC1, is really refractory to PD-1 therapy. But what's really, really apparent is that when you have that TET-on Th1 response and the endogenous CD4s and CD8s come along, now we rescue anti-PD-1 therapy in a model that was previously unresponsive. And if we do this in a kind of an easier model like B16, you don't even need PD-1 therapy to have a much better anti-tumor response, right? Just the presence of the TET-on TBET smartest, I even think we did 100K for this experiment, is just sufficient to drive both endogenous Th1 differentiation and effector CD8 T cell differentiation. Right, and so from that experiment, we know that this transition from stem to Th1 is really sufficient, and I want to point out it's in the presence of Tregs. It's sufficient to undertake that restricted program and drive effector differentiation of stem-like CD8s to effectors. How is this happening was our next question. So what is the mechanism by which this differentiation is driving a better anti-tumor CD8 response? And so from the single cell RNA-seq analysis I showed you before, we also sequenced the CD8 T cells, and we did this niche net prediction analysis that can predict ligands and receptors that are probably interacting within this Treg depleting compartment that drives effector differentiation of CD8s. One of the primary hits, there were a lot, but one of the main hits was interferon gamma, through the interferon gamma receptor and stem-like CD8 T cells driving this response. And so we did a very crude analysis to see if this hypothesis might be true. And so we blocked systemically interferon gamma in the presence of diphtheria toxin. So we selectively deplete Tregs and blocked interferon gamma, or just selectively deplete Tregs. And as you can tell here, right, just by blocking systemically interferon gamma, this was sufficient to abrogate completely the beneficial anti-tumor response. And so you could argue here, right, interferon gamma can target many, many different cells and has many functions in both innate and adaptive immunity. And so we really wanted to get at where is interferon gamma acting intrinsically and where is it coming from, right? Because we could also argue that maybe NKs or CD8 T cells themselves are producing interferon gamma. And so the first question we said was, where is it coming from? Is it the Th1 CD4s that are required? Is it the Th1 CD4s producing interferon gamma that are required to drive this process? And so how we did this is we either transferred wild type TET on TBET SMARTAs that I just described, or TET on TBET SMARTAs that were interferon gamma deficient. So they can become Th1s, but they can't produce interferon gamma once they're in the system. So how we did this was through electroporation of two different guides. We did this into an established tumor, followed by doxycycline, which turns on the TET on TBET program, and anti-PD-1 like we did before. And this is just to confirm that the interferon gamma knockout worked. And so wild-type TBET SMARTAs produce a lot of IL-2 and a lot of interferon gamma. Interferon gamma knockout, although they produce a lot more IL-2, they can't produce interferon gamma, and they can still differentiate into Th1s. And what we find within the endogenous CD8 T cell response is that when TET on TBET SMARTAs are present, and this is just a repeat of the previous experiment, we start undergoing effector differentiation within the draining lymph node. We get a huge tumor effector response. When these cells can't produce interferon gamma, this response both within the draining lymph node and primarily very dramatically within the tumor gets abrogated. And the anti-tumor response itself is fully abrogated. So these mice are no longer responsive to PD-1 if the Th1 CD4 T cells can't produce interferon gamma, really suggesting that it's coming from the Th1s. Right, and so now we can start filling in our little model here of the active response. So both the differentiation of stem-like CD4s to Th1s is important, as well as the production of interferon gamma from these cells that is acting on the response. Now, interferon gamma, as I said before, can act on many different cells. And so we wanted to see if this was a direct link of interferon gamma from the CD4s intrinsically on the stem-like CD8 T cells. And how we did this was also through a bone marrow chimera system. We developed a inducible interferon gamma receptor knockout CD8 T cell. And so these are P14s. So these P14s are TCR transgenic CD8s that are specific for the GP protein within these tumors. And we can inducibly knock out the gamma receptor also with the presence of doxycycline. And so this is just a confirmation of the knockout after doxycycline. Well, how we did this was we transferred either wild type or inducible knockout P14s. We actually waited for about 4 days for these cells to activate and establish normally, because interferon gamma receptor on its own at the beginning might be important for the priming, so we didn't want to mess with that. But after 4 days, we gave doxycycline to inducibly knock out the interferon gamma receptor, followed by Treg depletion to see if these P14s themselves can't sense interferon gamma. Do they still undergo effector differentiation? And what we found is that when they don't receive interferon gamma signaling intrinsically, these cells can't undergo effector differentiation like their wild-type counterparts. as a comparison, right, the endogenous GP33 response, right? So in the environment, the other cells that can sense it did undergo effector differentiation similarly to the mice that had wild-type counterparts. So really, really indicating that it is intrinsic to the CD8 T cell to sense interferon gamma to drive this effector response. And so now we can fill in a lot of this model and what we think is happening within this anti-tumor response, particularly within the draining lymph node itself. And so we have, we characterized both within the mouse and the human, the stem-like CD4 population that's really, really apparent within the draining lymph node. They activate, they fail to acquire this canonical program, and it seems like within the tumor, it's more prone to undergoing Treg differentiation, which through mechanisms that we don't know is restricting both a stem-like CD8 and stem-like CD4 T cell response. Now, we can switch this restricted state by targeting the stem-like population. We showed that in the presence of Tregs, if we inducibly force some antigen-specific CD4s to undergo Th1 differentiation, these cells can bring their buddies along and also drive effector differentiation of Th1s from these stem-like CD4s that then through interferon gamma helps stem-like CD8 T cell response to drive an effector program within the draining lymph node itself that goes to the tumor and really enhances and rescues both the anti-tumor response and response to immunotherapy. Right, and so this is great in a mouse, but is this ever a possibility in humans? Can we find patients where this restricted program that we mainly only have stem-like CD4s and 8s is broken? And can these stem-like CD4s actually undergo Th1 differentiation in a patient tumor? So we went back to this is about 150 kidney cancer patients, and we found that in about 10% of these patients, there is a very big Th1 response. Over 40%, in the ones that are there have this very, very large Th1 population. And so this is just to show you what it's like when it's actually present in these few patients. Your average patient looks like this left column over here, which is what I showed you in the beginning. Lots of stem-like CD4 T cells, a large population of Tregs within these tumors, and very, very, very few Th1 cells that are TVEP positive. In this 10% of patients, when 20 or 30% of the cells are Th1s, they fully downregulate TCF1. They have a much smaller Treg population within their tumors. These cells are fully granzyme B positive. They've downregulated CD28. They're producing interferon gamma. And in fact, those patients, we didn't have a lot, but the 12 patients that we did have that had a large Th1 response within their tumors, they respond extremely, extremely well to immunotherapy. This is both a combination of PD-1 alone, CTLA-4, or a combo of PD-1, CTLA-4. And every single one of these patients that did have a Th1 response are capable of much better response to immunotherapy. And if we look at the actual immunology and we look at the CD8 T cells within these patient tumors, not only did they have better CD8 T cell infiltration, they also have a lot more effector CD8 T cells within their tumors. And within their draining lymph nodes, which we also had a few, we have a lot more effector CD8 T cells, which normally is not the case within patient tumors. And so it is in fact possible. And what I kind of want to conclude with is we have a very large population of this stem-like CD4 T cells that is within the tumor, usually within niches within the tumor, very close to CD8 T cells. And what we showed with the mouse data is that we can target this population, right? It is waiting for the right signals to actually undergo good canonical CD4 differentiation. And so maybe the next set of therapies is really targeting this TCF1 CD4 that is malleable and targetable and can actually, when it does undergo the proper differentiation trajectory, it really pulls the anti-tumor response together and benefits the CD8 response to actually do the right thing. All right, conclusion, I think I've said most of this, so in the interest of time, because I do want to get to your questions, I will just go to the acknowledgements. So I really want to thank my previous lab, Hayden Kissick, for all the training. It was really an amazing experience to be in his lab. Anyone that's looking for a postdoc, he's an amazing boss and fantastic lab. All of my lab mates, and particularly the two co-authors that really helped through this, Natalia and Evelina, were truly amazing through this. And we had a lot of help from the Ahmed lab as well, both for LCMB and a lot of different techniques. Of course, all the doctors and patient families for giving us the samples, and the flow cytometry core for all of their help. And with that, I'll thank you, and I'll take any questions that you might have.
Thanks, Maria. It's a great job. I really like it. It was a lot of job. We'll continue with the questions and answers. Remember, you can write your questions in the chat, or if you prefer, you can open your microphone for questions.
I think there are a couple here. I don't think I can. Oh yeah, I can open it. Okay.
Somebody wants to ask a question now. Well, I have a question.
Yeah.
Maybe it's basic. It's a basic question.
Sure, it's not. Go for it.
Do stem like CD4 T cells have the capacity to self-renew? I just do this because when you isolated PD-1 positive, TCF1 positive CD4 T cells and incubated in vitro in Th1 conditions, almost all CD4 T cells became Th1 cells. And practically there weren't any